Hello, students. A very good morning to all of you. Hope all are doing well. Please stay home, stay safe. Please build up more hygienic conditions at home. Have proper food on time. Don't risk yourself by unnecessarily going out. So, by saying this, let me start today's very important session, a basic session in genetics, that is recombinant DNA technology. Let me share my whiteboard with you and start explaining to you regarding this. Now, students, all of us know that exchange of genetic information between DNA segments of same species is termed as genetic recombination. Genetic recombination. The advancement of technology, one can transfer genes of one species to another species artificially. In this class, you will find out fundamental details of artificial method called as recombinant DNA technology, even it is called as rDNA technology, and gain insight into the steps involved accordingly. The technology of recombinant DNA was developed in 1973. Please remember, students, it was developed in 1973 by Boyer and Cohen. Now, let us see the question exactly what is recombinant DNA technology. Please listen carefully, students. It is a technology to produce an artificial DNA molecule by combining two or more fragments of DNA which are not necessarily associated with each other. Such DNA fragments are obtained from biological sources. Students, I repeat again, I'll write it for you. It is the technology to produce an artificial DNA molecule by combining two or more fragments of DNA which are not necessarily associated with each other. Several steps are followed to recombine DNA segment, students. Furthermore, under a proper situation or an ideal situation, a recombinant DNA molecule can replicate by entering a live cell. This said technology is also known as genetic engineering. This is also known as, famously known as, genetic engineering. In a broader sense, it is created through three different methods, that is transformation, non-bacterial transformation, and phage introduction. Let us find out these methods and explain them to make you understand more easily. Here, let us see what are the goals of this technology, why it is important. The importance of this rDNA technology. Isolation and characterization of genes. First, isolation and characterization of gene. Second point, desired modification in isolated gene. Desired modification in isolated gene. Third, artificial synthesis. Artificial synthesis of new gene. Fourth, modification of organism's genome. Fifth one, interpretation of hereditary diseases. Interpretation of hereditary diseases and their cures. Last one, enhancement or improvement. Enhancement or improvement of human genome. These are the few important goals or uses of this technology. This is what the priority of given in recombinant DNA technology. That is, first one, isolation and characterization of gene properly. Then the desired modification is done in the isolated gene. Artificial synthesis takes place in the isolated new gene. Then modification of organism's genome once it is inserted into a living cell. Interpretation of hereditary diseases. They are finding out what kind of hereditary diseases are evolved and what are the cures and treatments. Finally, improvement in human genome as well. Now, let us see the process of this rDNA technology. In brief, process of rDNA technology. The first process is isolation of DNA. DNA is isolated in pure form. It is isolated in pure form, which means they are devoid of any other macromolecules. The second one, cutting of DNA. For this step, the restriction enzymes are very important. What are important? Please listen carefully, students. Restriction enzymes are very, very important. It helps to identify the location wherein the designated gene is introduced into a vector genome. The said reaction is known as restriction enzyme digestions. Restriction enzyme digestions. The third, amplifying the DNA. Amplifying the DNA. Copies of genes are amplified through PCR, polymerase chain reaction, or polymer. It is essentially a process to increase a single DNA copy into several copies, that is, several thousand copies, after the desired gene of interest is cut with restriction enzymes. That happens in amplifying of DNA. Then, fourth point, joining of DNA. What happens here? The vector and a section of DNA is joined in this step. It is achieved with the help of an enzyme, DNA ligases. DNA ligases. Then last step we have insertion of rDNA into the host, or whichever host is required, you're inserting. Yeah. Here, rDNA is added to recipient host cell, and the entire process is called transformation. What it is called? Transformation. Post insertion, the recombinant DNA multiplies and manifests as manufactured protein under favorable conditions. These are the processes involved in rDNA technology. Now, students, let us see few applications of this rDNA technology. What are the applications? To produce recombinant vaccines. To produce recombinant vaccines. Then producing human insulin to facilitate better crop production. Better crop production. It for producing growth hormones for producing growth hormones in humans to treat dwarfism. for better gene therapy, for better gene therapy, to acquire DNA fingerprinting, to acquire DNA fingerprinting, to diagnose several diseases, to diagnose several unknown diseases. Now, after seeing this, let us see briefly about gene cloning in rDNA technology. Typically, a clone is defined as cluster of individual cells which come from progenitor. A clone is genetically similar with its parent cell when it replicates. DNA cloning is initiated when DNA fragments are inserted into DNA molecules. The set replicating molecule is the carrier of DNA vectors. Here, plasmids, E cell, viruses are among prominent vectors of recombinant DNA technology. So this is a brief about recombinant DNA technology. In my next video, we are going to learn in deep about this rDNA technology. Thank you.